Most of the mRNAs targeted by RIB-B encode iron storage proteins. Free iron is required by the cell under certain circumstances, but high levels of iron are toxic for the cells. RIB-B regulates the levels of free iron by controlling the level of iron storage proteins. RIB-B is expressed from a promoter recognized by a special sigma factor called sigma E. It is just like sigma S, which is a stress response factor as we discussed earlier. Expression of the gene encoding sigma E is itself regulated by RIB-B, and thus this sRNA is part of an auto-negative regulatory loop for sigma E. तो ये खुद ही जो है अपनी रेगुलेशन को कंट्रोल कर रहा है, जिसे हम कहते हैं ऑटो नेगेटिव रेगुलेशन. The stationary phase sigma factor, sigma S, is encoded by the RPOS gene of E. coli. Translation of RPOS mRNA is stimulated by two sRNAs, that is DSRA and RPRA. Activation is achieved by a switch in alternative RNA base pairing. The small RNAs bind to a region of the mRNA that otherwise would pair with the ribosome binding site, which thus inhibit translation. The RPOS gene is also acted on negatively by another small RNA, which is OxyS. Other regulatory RNAs act simply as antisense RNAs. They are encoded by the strand opposite the coding strand of a gene and act through homologous base pairing to inhibit expression of the mRNA produced from that gene. तो ये जो ये हैं, ये एंटीसेंस आरएनएज हैं, यानी कि जिस डीएनए स्ट्रैंड से ट्रांसक्रिप्शन हो रही है, ये उसके ऑपोजिट स्ट्रैंड से जो है वो सिंथेसाइज होते हैं और फिर इस तरह से वो बेस पेयरिंग करके कंप्लीमेंट्री हो जाते हैं और वो बेस पेयरिंग करके इसको इस एमआरएनए के एक्सप्रेशन को इनहिबिट करते हैं. These tend to be associated with genes encoding potentially toxic products and also in regulation of some phage genes, as in lambda. These RNAs are often said to act in cis because they act only on the gene from which they are made, which is opposite to the trans-acting sRNAs, because trans-acting sRNAs act oppositely. Let us consider other examples of gene regulation mediated through alternative RNA pairing that truly operates in cis. These are RNA regulatory elements that control expression of the genes within whose mRNAs they reside. So, जिस mRNA में ये reside कर रहे हैं, उसी के expression को ये control करते हैं. The most striking example are the so-called riboswitches that control metabolic operons and attenuation in biosynthetic operons. The TRP genes of E. coli are the classic examples of the later mechanism and are where RNA-mediated regulation was discovered.